Okay, Mark, I am about to hit start webinar right now.
Good afternoon and welcome back to day 2 of our public meeting on evaluating real-world evidence from observational studies and regulatory decision-making, lessons learned from trial replication analyses. This webinar is being convened through a cooperative agreement between the Food and Drug Administration and the Duke-Margolis Center for Health Policy. I'm Mark McClellan. I'm the director of the Duke-Margolis Center for Health Policy at Duke. Yesterday, we heard about a target trial approach to trial replication or emulation from Miguel Hernan, and 3 presentations of trial replication projects that sought to better understand the strengths and limitations of observational studies for regulatory decision-making through the target trial type lens, the study replication lens, as much as that is feasible in observational analyses. The researchers who are leading these trial replication projects, and there are now many of them, shared some of their initial findings. They found that in many cases, the results of analyses of secondary real-world data agree or comport similarly to the results of the clinical trials. They highlighted that agreement between the randomized controlled trials and the real-world evidence depends on a variety of factors, including the similarity of the underlying populations, the use of active comparators, and paying attention to details in setting up the observational studies that made them like the clinical trial conditions. They also talked about some of the challenges related to using real-world data, like outcome ascertainment and validation, and the fact that there's not really a exact equivalent for placebo groups, for example. Panelists also discussed some of the considerations for mitigating bias and adjusting for confounding when using real-world data from claims and from electronic health records, keeping in mind that it's not just about randomization. It's also about data quality and completeness, fitting in with the FDA's framework for approaching real-world evidence. We heard about the importance as well of transparency and pre-registration of study protocols for building study credibility and making them, again, more similar to the standards in place for clinical trial data that is used for regulatory and clinical decision-making purposes. Now, we're going to continue to build on a lot of these topics during the discussion today. So here's our agenda. We're going to have 2 sessions of panels to build on the lessons learned from the trial replication projects that we heard about yesterday. Session 4, the one coming up first today, will focus on the types of study questions that may be appropriate for consideration through observational studies, including which endpoints can be identified using real-world data and where additional methodologic improvements are still needed. Session 5, our last session, will look at next steps for understanding the potential role of observational studies in contributing to evidence of drug product safety and effectiveness. The panelists in this session will also seek to identify some of the remaining gaps in knowledge and strategies and practical next steps for addressing those gaps. So just a few reminders before we get started. First, you can visit the Duke-Margolis event website for the materials from today's webinar. And that includes recordings that will be available after the event for people who missed it or want to go back and hear some details again. If you have any questions for the panelists, we encourage you to participate. We got a lot of really good questions that livened up the discussion yesterday. You can submit those questions via the Zoom Q&A function at the bottom of your screen. Because we are recording, we want to remind our speakers to please check that your camera and microphone are active before speaking. If you have any difficulty for our speakers and commenters, please remember that we can help you remotely by activating or deactivating your microphone. For the presenters, just a reminder that Valerie Parker from the Duke-Margolis team will be advancing the slides today. Please give her a verbal cue when you're ready to move to the next slide. In addition, Mira Gill is going to be providing some timing reminders to help us keep on schedule. So for the speakers, for the panelists, look for notes from her coming through the Zoom chat function. And for all of you, I encourage you to join us at Duke-Margolis on Twitter. That's at Duke-Margolis, all one word. And you can use the hashtag trial replication. So hashtag trial replication. And we invite you to follow the Duke-Margolis Twitter account and join the conversation online as well. So again, we're very pleased to have you with us today, and we're going to get right into our first session on key themes emerging from replication efforts. So to kick us off, we've got some very experienced experts who are going to pull out some key themes from the replication efforts that we heard about yesterday and have been going on up until now. Our panelists include Lucina Orsini, who is Vice President for Value and Outcomes at Compass Pathways, Josie Briggs, who is Senior Advisor at the Patient-Centered Outcomes Research Institute, Michelle Johnson-Funk, who is the Director of the Center for Pharmacoepidemiology at the UNC Gilling School of Public Health, and Robert Ball, who serves as Deputy Director of the Office of Surveillance and Epidemiology in the Center for Drug Evaluation and Research at the FDA. So first, we're going to turn to Lucinda. So over to you.
Thank you, Mark. I'm really happy to be here, and thank you for inviting me. Valerie, you can go to the next slide. So like Mark said, I'm Lucinda Orsini. I am here, in this case, representing also ISPOR. So I was the former Associate Chief Science Officer of ISPOR up until a few weeks ago. So I'm happy to be here on their behalf as well. Valerie, you can go to the next slide. So just in keeping with the idea of what themes emerging from yesterday's discussion, which I found really great, I think, we all agree that there's, some key ingredients that go into making real world evidence useful and of high quality. And we talked a lot about some of those yesterday, and they're certainly coming through in the discussion around replication and reproducibility. So quality production can't be, understated in any way. Careful data collection and curation is of utmost importance. The appropriate analytic methods, I think, came through loud and clear about how important those are. Good procedural practices for transparent study processes are also important, as well as the replicability and reproducibility that really can highlight, the quality of these data, especially if they're handled correctly and analyzed appropriately. And I thought the target trial, Dr. Hanan's target trial ideas are really thought-provoking in the sense that we're really hoping that when people pull together a real-world evidence study, that they're setting up according to the research question, finding the right data, the right analytic methods that are going to answer that question, and being very transparent about the underlying study processes and the data itself, so that we can have responsible consumption. And that's one thing we didn't talk too much about, but I wanted to touch on here, and maybe we'll talk a little bit at the end of my remarks as well, that we really need informed interpretation and fit for purpose application of these types of real world evidence studies. And that really can't be sort of underemphasized, because I think there was a great example, I think, that Dr. Hernan gave about a really, excellent study that was done, but wasn't even published because people really didn't understand exactly how the methods and the biases were, accounted for, or the editorial board or staff itself didn't see the value in the study in publishing it. So we really are up against a couple of different factors here, both on the production side and the consumption side. Valerie, you can go to the next slide. So, how important is it that real world evidence replicates, I should say replicate RCTs. So I know that this is what the whole 2 days is about, but we have to remember that when we're using real world evidence, we're often using it to answer a question that a clinical trial either can't answer or didn't answer. So we generally don't think of real world evidence as replacing a clinical trial, but as additive evidence too. We know RCT endpoints can't always be recreated in the real world, and they're not collected in the same way, and don't always have a lot of the similar meanings in clinical practice. That patient populations can't be completely recreated, as we've seen in some of the examples from yesterday. Prescribing patterns aren't the same in the real world and often don't follow those label indications. And we usually need to combine data sources to get a full picture. Claims, EHR, primary, secondary, inpatient, outpatient, lab, and test data often need to be combined together in order to really get a robust sense of where we are. And many times the real world evidence isn't aligning with the clinical trial evidence, and what then? Valerie, you can go to the next slide. It just highlights that last little box at the bottom. So we're talking about, here replicating clinical trial evidence with really high quality studies that are designed to do so. If you go to the next slide, Valerie, this is just a poster we presented at ISPOR that we're writing up now for probably a peer review publication. So take it for what it's worth. But we did a review of systematic reviews that had evidence of both clinical trial and observational. So these are reviews, and we use a similar search strategy to Engelmeyer, which you can look here in this publication. But we looked at papers between 1990 and 2020. We focused on pharmaceuticals only, and we used search criteria that were looking at reviews that were designed to compare relative treatment effects of pharmaceuticals from observational studies and had corresponding effects from clinical trials. And so these are wild type observational research, things that are out there that weren't necessarily designed to replicate. And what we found is that there is variation in the consistency between observational and clinical trial treatment effect estimates. So, about 20% of the time, there is a statistically significant difference between the relative treatment effect that you're finding between these two types of studies. So we do need to be a little bit careful in thinking about what happens when observational studies are not necessarily replicating a clinical trial. And does that mean that the observational data is wrong, that we can't use the evidence? I mean, I think that's not necessarily what we're thinking about here. But in order to make a qualified judgment about the real world evidence, we need to be transparent about what was done. So Valerie, you can go to the next slide. One thing that we've been working on at ISPOR and actually in collaboration with Duke-Margolis, International Society for Pharmacoepidemiology, and the National Pharmaceutical Council, among others who you see listed here as authors on this paper, is pulling together recommendations of how to be more transparent with our real world evidence studies, particularly these secondary data use studies that are looking at comparative effectiveness. And I think, by showing an example, you can go to the next slide, Valerie. We saw some of these studies like operand and RCT duplicate actually, using good study hygiene. So they are trying to be as transparent as they can, actually registering these studies and their protocols on either clinicaltrials.gov or EU PAS. So, there's ways that we can be more transparent so that people can really understand exactly how a study was done, whether those results can be something that is applicable to a decision that they're using or have at hand, and that we're not just here to only replicate a clinical trial. You can go to the next slide, Valerie. And the reason this is important is because we know that basically real world evidence has a public relations problem. We saw the retractions of these high profile studies that were done with the Surgisphere data on COVID-19 endpoints or products that were important for COVID-19. Rightfully so, they were retracted, but it was because we didn't understand how the studies were done or didn't have access to the underlying data, and that was a problem. But we also know that people are fearful that real world evidence is going to, or we're trying to replace clinical trial evidence, and that is also not necessarily what we're trying to do here. So I think these were just some of the themes that came out yesterday, and some of the things that I think are important to discuss going forward. So I'll leave it there. Thank you very much.
Lucinda, thanks very much for that presentation. And next is Josie.
Thank you, Mark. Do you hear me?
Yes, just fine.
Great. So it was a wonderful afternoon yesterday. I learned an enormous amount. I've been following the trial emulation work with a lot of interest. We enjoyed having Miguel Hernan visit us at PCORI and present to us. And I have found this whole body of work very stimulating and thought-provoking. Can I have my first slide, please? Next slide. Yeah, next. But at the same time, there was a certain amount of journal editor bashing yesterday, and I have to admit, I'm an editor-in-chief of JASON, the leading nephrology journal, and I have to admit I would be guilty as charged with some skepticism about treatment comparisons based on observational data. But I am coming around to believe that these tools and this really brand new approach to observational work can really build substantially more rigor into the examination of treatment effects. A telling example that Miguel used yesterday was the IL-6 antibody studies. And the point he made in that presentation was that all the known biases would have worked in the direction of not capturing benefit, but the observational data suggested benefit. I thought that was really an important insight, and it's how in the future studies I see as a journal editor, I will try to start with saying, well, what might the biases be, and what direction do they go in? I do think that the emulation work is really setting a new bar for high quality observational work, and that approaching observational studies with really the kind of mindset that a clinical trialist has, we will get much solider answers. But I think it's also important to acknowledge, again, some of the limitations these approaches are going to have. Most of those came up yesterday, but I thought I would just highlight a couple. The first concern often is, are the initiator and the non-initiator for the intervention truly matched? And I will give you a counterexample that we've been struggling with in the journal I edit, which is, does remdesivir cause acute kidney injury? As I'm sure most of the listeners know, remdesivir is being used very extensively in most hospitalized COVID-19 patients right now. And many of these patients sick enough to be in ICUs develop acute kidney injury, maybe 50% with maybe 15, 20% require renal replacement. When one compares, even pretty carefully propensity matched, patients who get remdesivir and those somewhat outliers who don't, remdesivir is associated with more AKI. Is it causal? There's a biological plausibility here because of some of the products of remdesivir, but I would say we really don't know. And I'm skeptical as to whether observational approaches will ever solve that kind of problem. Another thing that was discussed extensively yesterday is the outcome measures. The limitation of outcome measures in heart failure was discussed quite thoughtfully. Ascertainment bias, particularly with electronic health record data, but also claims data, can have an important source of bias or skewing of results. And I think that's something that has to require continued attention. A classic example close to my heart of where ascertainment is very uneven is when is serum creatinine measured. Serum creatinine is measured a lot in a lot of clinical interventions, but it is more often measured when people have volume disturbances. So you end up with a bias that may suggest that your two groups aren't perfectly balanced. So I think these emulation techniques are really important. I think it's really going to raise the standards for observational work. And I really enjoyed and learned a lot from yesterday's presentation, and I'm eager to see these methods become more broadly applied, including application in kidney disease. I will add that I think we need more help of some of the things that began to be discussed yesterday. There was discussion of data sharing expectations. We at Jason have worked hard to set high standards for data sharing, but I don't really know how to make this work for this kind of work research, and I'd love to hear from other participants about that. The pre-registration issue, which Mark mentioned, is important. Pre-registration has been incredibly valuable way for quality assessment in clinical trials. The first place we look is clinicaltrials.gov as to what was actually the pre-specified primary outcome variable. But pre-registration, as Miguel outlined, is a little more complicated in this setting. So I'm interested in hearing more thoughts about that. I'm also interested in what people can tell us how we guard against publication bias. How do we think about, I think publication bias is much less problem with RCTs. Well, it's still the problem of whether people actually write up the studies, but when they come to journals, I think we're all very comfortable in publishing negative studies. But that may be less so with this kind of work, and I think that's worth talking about and hearing advice on. Next, one more slide. I also have one other perspective on this interesting discussion yesterday. I spent a good part of my time of my career as a public sector funder. And one of my, although the purpose today and yesterday of the discussion is how can real world evidence emulation studies contribute to the regulatory decisions, I think they have enormous interest and potential for public sector funding as well. I was thinking as I was listening to this, that the next time I'm in the position to push forward an RCT, I would like to have in my pocket a pre-emulation study so that we could duplicate the study that we're expecting to fund with a observational study that would ask some of the questions I've listed here. And certainly one of the things that really is important in public sector funding is learning what doesn't work. And so I think this will be an important application of observational work. And I finally want to just end with one small worry. When you're a funder sitting either at the NIH, PCORI, I'm sure at VA, it's a very risky business starting a large, expensive randomized trial. There's lots of reasons why people get hesitant and advisory councils and boards get hesitant. So I do want to interject a small worry that we not stop telling the world that we still need the gold standard trials, that the emulation studies are going to broaden our evidence base. Perhaps they're also going to help people who write guidelines, but the gold standard trials remain absolutely critical. Thank you very much.
Josie, thank you very much as well. So we appreciate that. And now on to our next commenter, Michelle Johnson-Funk.
Thank you. Next. So I would like to start out by highlighting something that I think is a really important issue for us to recognize in sort of the discussion of how randomized trials and real world evidence may tell us different things. So what we heard yesterday is that the duplicate effort, as well as others, has really focused in on dialing in on and eliminating the reasons that the real world evidence result that they produced might differ from a randomized trial. But there are many reasons that the results from a real world study and those from the trial might differ outside of the duplicate efforts without implying that the real world evidence result is biased. And recognizing that, I think, is a piece of being able to then identify the value of what we can learn from the real world evidence and to not immediately assume that that result is incorrect when it is not the same as something that we previously saw in a at least superficially similar trial setting. So next, I know that we discussed the possibility of random error simply accounting for the difference in the estimates or the precision around those estimates being really different. So we've seen examples of the real world evidence studies actually having much greater sample size and therefore more precise estimates that might, at least on the surface, imply a different answer. Next. We also know that these might actually be answering different questions, that the estimand for the trial population may not be the same as the estimand in the real world evidence study if we're focusing on, for instance, a treatment effect in the treated versus the treatment effect in the total population. Next. Treatment effect heterogeneity is also a really important source of potential true differences. And I just want to note here that if the real world population that we're studying this question in has a different background rate of the outcome that we're interested in, and the treatment does have some effect on it, then we're guaranteed to see treatment effect heterogeneity on either the absolute or the relative scale if the populations differ in that sense. So again, this is not to say that the result in the real world data is wrong. It's actually an accurate representation potentially of the effect in that group. And it's simply that the background risk in those individuals is different than what we saw in the trial itself. Next. Another possibility that I think we're going to be coming to grips with, certainly in COVID, is that the treatment itself is different potentially in some important way. So within the trial setting, we might have a highly controlled dosing interval for 2 doses of a vaccine. And in the real world, we are delivering those doses at more variable intervals. So we might see different treatment effects that are due to true differences in how the treatment was provided. Next. I know we discussed a lot about outcome differences, potentially in the duration of follow-up, and that hazard ratio is going to be some average across the follow-up time that was used. So if there's some variation over time in what that hazard is, we're going to see an average of it, and we might be averaging 3 years in the real world data instead of 18 months in the trial. Similarly, competing risks can be an important influence on how many of these outcomes we observed. And this is something that real world populations can differ from trial populations in fairly substantial magnitude. Next. And my final note here is something that I know is discussed in the context of the target emulation approach, that adherence in real world populations is likely to differ from adherence in trials. And just my note here is just sort of a thought experiment. You can imagine identical prescriptions, identical populations, but the costs associated with the prescription might differ. And that could lead to differences in adherence. And now this is getting to what is the effect that we're trying to estimate. And if it is more of a pure biologic effect of the drug, we can then analyze that in a way that we and follow up if patients who are not fully adherent. But just to be aware that these are all reasons why we might get a different answer, and it's not wrong per se. Next. So next, I'd like to go through sort of my take-home messages, and both Josie and Lucinda have highlighted a number of these, so some of them I'll pass over quickly. Next. First, data quality is really key, and recognizing when the data are not sufficient to support the analysis means that you have to have a deep understanding of the data source and the context in which it was generated in order to know that. And those skills are arguably a little bit different than the skills involved in evaluating data quality in a randomized trial. Next. And another issue that I often hear when I'm talking to investigators about study design is they don't want to throw away people. We have observations on individuals here who were using the drug that we're interested in studying. We don't want to throw away that data. The truth is that this concern about loss of sample size shouldn't justify including individuals who are not appropriate for the target trial that we're interested in. If we, as Miguel showed us, we can find a wildly unrealistic protective effect of, say, statins on cancer, by including a lot of prevalent users. And that is not going to help us, even though it's going to improve our sample size. Next. This was discussed many times, and I think it's worth saying again that a fancy statistical analysis cannot make up for fundamental errors in study design. And we talked a lot about appropriate identification of time zero yesterday. I think of these as problems of use of crystal balls and time travel in epidemiologic studies. When we have access to all of the data, it's very easy to pull out the crystal ball and look forward in time to see what is going to happen to these patients after time zero, and to start mucking with how we define them as exposed or not based on looking into their future. And this is not something that we could do with a patient sitting in front of us in a clinic who we're making a treatment decision for. So if that information isn't something that you could know at time zero when you're interacting with a real patient, you can't use it to define these aspects of your study. Time travel is sort of the opposite problem. It's where we imagine that we could go into someone's past and change their last 4 years of experience. So if we're comparing prevalent users of drug A to prevalent users of drug B, we can't go back in time and switch those two groups, because we can't give the people in drug B 4 years of history with drug A by moving them over. So if you have to invoke time travel or use a crystal ball to envision how this study would work out, that's a good clue that you're in a problematic area. Next. There are a number of key design features that we've talked about: active comparators, appropriate use of time zero, critical aspects of how we define follow-up and measure that. And these are somewhat different than the design features that we use to limit bias in randomized trials. So I think Josie told us to sort of bring a trial mindset to a study, but you also need a pharmacoepidemiologist's training in the specific study design approaches that are needed to limit the bias in these particular non-randomized, non-interventional studies. And to go with that, we need individuals with that skill set who are able to provide reviews at major medical journals, major epi journals, to make sure that the results that appear in publication are as robust as possible. Next. And finally, I think we heard over and over again that although there's a lot of worry about unmeasured confounding, it tends to be less problematic than other sources of bias. That the biggest problems arise from things that are upstream from the moment when you start to worry about unmeasured confounding. That said, we can't ignore it. Next slide. So I want to highlight some high-level findings from ongoing work that is funded by the FDA. This is another demonstration project, and this project is called DETECT, Detailing and Evaluating Tools to Expose Confounded Treatment Effects. And the first portion of this work was focused on really looking around to see what tools are out there and how are they being used now. And the bottom line is that the worry about unmeasured confounding is common. In a systematic review of papers published in major medical journals or major epi journals over the 3-year period, almost all of them, 90%, listed some worry about residual, unmeasured, or uncontrolled confounding as a limitation. So great, we're all aware that we should be thinking about this. Next slide. The bad news is we're really not explicitly assessing the threat. We're not doing the work of using the tools that we have at hand to quantify and characterize the direction and magnitude of the potential bias and what that means for the conclusions. So even of those articles that said, yeah, we're worried about unmeasured, uncontrolled confounding, 43% of them didn't actually do any sensitivity analyses to assess that. And of the ones that are doing something, they're not using the methods that I think we would consider the best practice, which is to quantitatively apply something to adjust that estimate and give us a better sense of the uncertainty around it and where it may actually lie. So I will close with that. I think this is really exciting work, and I think it's put us on a very solid footing to take the next steps in the use of real-world evidence to try to understand drug safety and effectiveness.
Thanks very much, Michelle. Next, we'll turn to Bob Ball.
Thanks for having me. I'm going to focus my comments on the issues around data and specifically on outcomes. I think several people have already mentioned this, but we heard yesterday that in the RCT duplicate project, the trials that were selected for emulation were selected at least in part on the basis of their ability to have fit-for-purpose data. And despite that, there were at least two examples where problems with the measuring the outcomes in claims data might have contributed to the inability to replicate. And the two outcomes were heart failure and angina. And so those have been discussed fairly extensively. But what I found interesting about this was that in our experience in Sentinel, looking at safety outcomes for the last several years, we found that about 50% of the reasons that we were not able to use the Sentinel system for regulatory purposes in the context of the fit-for-purpose data assessment was because we couldn't find outcomes that met the need. And the reasons behind the outcomes was either we didn't have the specific type of data in claims, or because an algorithm that was used to assemble claims data into a computable phenotype didn't have adequate performance characteristics. So this suggests, I think, that this issue of outcomes and how we go about addressing this is going to be very important to further advance the use of real-world data, whether it's for safety or for efficacy evaluations. So when we first did the formal analysis a few years ago of this, we held a workshop with Duke-Margolis to discuss this further. And one of the results of that was to begin two projects to further address this, combining machine learning and natural language processing tools with electronic health record data. So we've recently completed those two projects. One was looking at rhabdomyolysis, which is a muscle injury that sometimes rarely occurs after drugs. And in that project, what we did was we asked whether machine learning methods applied to claims data only could construct a computable phenotype that was performed better than human experts alone. And what we found was the answer to that question, at least in the case of rhabdomyolysis, is that the answer was yes. And it still didn't get a perfect performance, but the fact that it led to improvement was encouraging. The second project we launched was around another complex clinical outcome, anaphylaxis, which is a allergic reaction that rarely occurs after drugs, but is important for drug safety evaluations. And in that project, we evaluated the use of natural language processing to extract information from the clinical text and electronic health records to construct features that could be combined with claims data to see if we then could apply machine learning to those variables to improve the performance of the algorithm. And there we found, again, that we were successful in getting an algorithm with improved performance. And then we took that to another level and we said, how will this algorithm perform in another data system? And we found that the algorithm performed surprisingly well although not quite as well as in the first data system. And this illustrates a very important point about outcomes in real-world data. And that is that there's a very strong desire, naturally, to have transportable, computable phenotype algorithms available. But it turns out that different information is represented differently in different healthcare environments. And so careful evaluation of the transportability of algorithms remains an important characteristic. And a lot of care, I think, will have to be taken as we move into this realm, especially for complex outcomes. Subsequent to these two projects launching, we developed a 5-year strategic plan for Sentinel, which includes more broadly the creation of a more easily accessible EHR data, along with a formal framework for applying NLP and machine learning to develop computable phenotypes that will be transportable across the entire network, or at least will give us an understanding of how one goes about doing that. So this is another general activity that I think others can also work on within their own systems. Just another note on that. So we've now launched several more projects, but there are novel approaches also to trying to look at the information in different systems, the way they're coded, and to see how they can be essentially translated as if you were translating one language from another. And this might be a fundamental advancement in the approach we take to developing computable phenotypes that are more generalizable. And we'll see how that project develops over the next couple of years. And lastly, I'll just mention another example from COVID-19. So as we've tried to do either our own studies understanding the natural history of COVID or evaluate studies of comparing therapeutics in COVID-19, one of the issues that has come up is that severity of the disease is often linked to the clinical use of oxygen. And it turns out that oxygen and how it's used is coded very differently depending on which healthcare system you're working in. And it typically isn't well captured in the structured data in EHRs even. So one has to take a deeper dive into the notes. And the question is, where exactly in the EHR? It might not be in the physician notes. It might be in nursing notes. It might be in respiratory therapist notes. So we're undertaking some validation exercises in some of the different systems we're working in to further understand that complexity. But it illustrates just another very important point about outcomes, especially when we get into complex clinical outcomes in hospital settings. And that is that each time we come across a new issue, it requires a fairly deep dive into the data. And some of the other panelists were talking about that idea that we really need to understand this data. And Bob Temple yesterday mentioned in his comments that for certain outcomes, we have more experience and we're likely to be more comfortable with using real world data. And that's probably true, but the real interest, I think, is in trying to understand how we can apply these approaches for novel outcomes. And that's going to require a much more sophisticated and systematic approach to developing sort of these approaches to using real-world data outcomes for these complex clinical conditions that don't necessarily require a very deep human effort, because otherwise it reduces essentially the efficiency of using these kinds of data. So there's a whole other, I think, effort that's going to be needed to bring real-world data use up to another level. So thank you for allowing me to make those comments.
Thanks very much, Bob, and great comments from all of you. To those of you who are joining us today, we did set aside a significant amount of time in this session for addressing questions or comments that you might also have or about the studies that have been presented and about the state of the art for some of these real world evidence activities that we just had a really great set of perspectives on from our panelists in this session. So don't forget to submit the questions in the Q&A box. For now, and this is picking up on some of those questions that have already come in, Bob, I'd like to stick with some of the data themes that you mentioned. We'll get to the points about with observational data, the concerns about potential bias and other issues as well. But as you said, a lot of these issues are kind of more basic, and there are a couple of questions in the that have come in picking up on the fact that we much of the studies that were presented yesterday were based on claims data, not really more extensive than that. Bob, you highlighted some of the limitations. I'm assuming that's particularly in claims data of being able to measure key outcomes, especially if there's a significant clinical component to that. I was wondering if you all could comment a little bit on what your current take is on the state of the art for using data and the best practices there. In particular, Bob, this gets to another of the comments about, you talked about the difficulty of sort of transportable computable phenotypes. Is basically, I'm translating that as a set of algorithms that could be applied to data, claims data or otherwise, in one healthcare system, one health plan or healthcare organization, and a similar one could be used elsewhere. It was interesting that yesterday that most of the results presented were within one system, whether it's Aetion or Optum or otherwise. You talked about how much work Sentinel has put into developing data standards and understanding. They're pooling across a number of healthcare organizations, but I know a lot of work has gone into making sure the data are consistently interpretable. Could any of you expand on where we are with those kinds of activities? Is there, at some point, can we think about really transportable phenotypes? Are we still going to be needing to pay a lot of attention to understanding each particular data set and how generalizable it is or how much it differs from others for quite a while to come?
This is Bob. Maybe I'll just start by reinforcing some of the points I was trying to bring out in my comments. So you alluded to the idea that there's been a lot of work done in Sentinel on this, and it's done at several levels. So there's the common data model concept, and one of the values of a common data model is that it gets everybody operating on the same page in terms of at least what the definitions of different variables are. As a kind of classic example of the value of that is laboratory data often has different units, and just having things in the same unit can be very helpful. The other aspect in Sentinel Sentinel's put a lot of work into is what is referred to as routine quality checks. And that's just, it starts at some very basic things like, is this data element consistent with what is in this field? But then it goes up into more sophisticated things by looking at, whether or not trends over time of a particular code have some sort of dramatic aberration that would suggest that it's probably not real, but some kind of change in coding or something else going on. So those are sort of some of the basic things, and they're very important when working in a distributed environment or across different data sites. The next level above that then is taking all that data and trying to construct a computable phenotype or an outcome from different codes. And there has also been a lot of work in Sentinel on that, going back to mini Sentinel. The classic approach to that has been human experts will review a set of charts and say, is this anaphylaxis? And then from that, there's a calculation of typically a positive predictive value of a particular set of codes leading to that outcome, and how good is that? So one of the other things that first steps, I think, in a sense, is establishing what metrics should be used for assessing the value of a computable phenotype. Positive predictive value is commonly used mostly because it can be calculated, but there are other metrics that might be better. And as things get more sophisticated, there might be some more value in thinking about that. So in a sense, where I think we are in Sentinel anyway, in general, is saying, how can we make that whole process more efficient? So rather than having to have human experts look through all these charts and find all the different pieces of information that are relevant, can we have more automated ways using natural language processing or machine learning? But there will always be sort of a human expert piece to it, but it's sort of about how do we make that more efficient? And the projects that I was alluding to, I think, demonstrate that we are now at the point where this is all possible. Many others have done, related work. So now the question is, how do we take all that and put it into a more standardized and systematic approach where things can be shared, and at least we're speaking a common language. And then the next level beyond that, which I think we're just starting to get into, is to understand how to calculate whether or not the information in one system represented by a certain code is the same as the underlying information in a different system. So I'll stop there.
Thanks. That's a great overview, and thanks for illustrating it so concretely with the progress and the work that's gone into getting Sentinel to where it is in terms of being able to combine data from a range of systems, and now, as you said, going beyond claims data to including electronic records too. The other panelists care to comment on this general topic of the further steps or best practices to assure that you're using the data appropriately in these real world evidence studies?
Yes, I'd love to. I have a few thoughts on that. So I think one of the critical dimensions of thinking about the computable phenotypes that we use and the performance in various settings, the sort of most potentially problematic thing is if that differs based on the treatment that you're looking at. So often when we conduct these validation studies to characterize the performance of a particular computable phenotype or algorithm, and then we transport that to a different setting, unless it happens to be that we've already studied this in a setting where the treatment of interest was a factor and we evaluated whether sensitivity and specificity differed by that treatment in particular, it may be difficult to even know in the new setting if we have similar degrees of capture for the outcome. So I think that's one key dimension where I fear that we're not going to quickly get to the place where we're going to be able to say, oh great, this algorithm has been validated, we can use it. The second reason that I think that is going to be an ongoing opportunity for work is that the relative importance of high sensitivity and high specificity differs depending on the setting in which you're using a variable. So generally speaking, we want highly specific outcomes for relative measures of effect. If we are more interested in an absolute treatment effect size, it actually may be more important to have a sensitive measure. So the application for that algorithm, the role of that variable in a particular analysis can actually change what kinds of performance characteristics we want. And then the last thing that I worry about somewhat is that the gold standard and the validated algorithms do rely on a human judgment about what the truth is. And there are places where our human judgment about the truth has already sort of been imbued with biases, racial biases in some cases. And if we perpetuate those under an umbrella of, well, we trained the algorithm and we can move on. And that concerns me a lot, that we might inadvertently perpetuate those if we don't take a close look at sort of the quality of the gold standard data that we're using.
That's a really good point, and that those biases do get worse if your underlying data isn't measuring exactly what you wanted to measure. If you're using proxies for health status that are instead utilization measures, for example. So I think that does get back to the core importance of really understanding the data well and making sure it's fit for purpose. Other comments?
Just to amplify a little, or to echo what Michelle said, I do think we are stuck with the need to validate in the setting of the specific research question because of the possibility that our algorithm may operate differently on the two comparator groups. And a second area of worry is we have healthcare systems that are operating very differently for different populations. I've been struck very recently at how segregated healthcare is in much of the US. We're seeing this in PCORnet data. And of course, it's really appropriate to worry about the huge disparities, health inequities in our healthcare delivery. But it also means that the settings in which we are extracting information will carry different kinds of levels of completeness and error. So that's part of why this kind of, yes, we need these algorithms. Yes, we hope they move across systems that hopefully are increasingly interoperable, but we've really got to check whether it's going to work for the question we have. And I guess my own bias, maybe this reflects my age, but we still need people to do that.
It is challenging. And Josie, I'm sure just a little bit of follow-up on that. I mean, you're having to live through this a lot with PCORI, where you're trying to support real world studies across a range of these settings and systems, including some that are serving typically underserved populations. Is PCORI finding some promising directions forward to addressing these data validity issues?
Well, I think yes and no. I think we're recognizing that they are critical, and it's really critical to PCORnet's drive to make large studies utilize real world evidence as a core piece of how they work. I do think that collecting information in settings in which there is a smaller institutional investment in the IT infrastructure is harder. And I think we have to go into this knowing that. It doesn't mean that the well-resourced systems always make the research question their highest priority. They've got other things to do. And so I think building the extraction of real-world evidence into prospective studies, randomized studies, and using it for trial emulation just is it's incredibly promising. It's incredibly important, but one can't forget that it requires the level of attention to detail that we heard yesterday, and that kind of attention has to be brought over and over again to questions.
Yeah, thank you. Lucinda, any comments to add related to these data validity questions?
No, I mean, it's one that we know is huge, and I think overriding in some cases, as some people have talked about, and one that's hard to sort of crack from a transparency perspective, because a lot of these data are protected by an IP, and it's not easy to get direct access to the data itself in some cases. And even if you do, you have to have a certain expertise in order to be able to understand the variables and how they were, put together into the algorithms, and just having access to the coding isn't always enough either. So it's really complicated. And I think it's something that we need to start working on directly with the data source owners as well, so that we can bring them into the fold of thinking about things transparently, because that is really important.
Yeah, thank you. Before we leave these, sorry, go ahead.
Can I ask if anybody on this illustrious panel or any of our participants have advice for journal editors? What should our data sharing expectations be in this kind of space?
Yeah, I was going to get to that a little bit later on how
what
are some of the steps to encourage this, but I think it's I think let's put a pin in that. I do want to get to it in terms of there are a whole set of issues, right, for journals around transparency, as some of you noted, were noted yesterday, advanced specification methods, et cetera. So I do want to get to that. Let me stick with one other data question for now. A couple of the questions in the chat asked about patient-reported outcomes. Now, these are not typically captured in claims data or electronic records and the like. They have to be, maybe they're reflected there. Josie, I know you've spent a lot of time on thinking about PRO data, but are there any particular special validation considerations there? Does that present a way of maybe getting beyond some of these platform specific issues if we've got well-formulated, validated PRO measures?
Well, this is a complex, many-faceted question. I do think building more patient-reported outcome measures into clinical practice is also an important direction that will contribute to eventually these kind of measures being part of care more often and being captured for some of the purposes that we've talked about. The PROMISE initiative, which I led for a number of years at the NIH, was, we thought when that was started 2 decades ago practically, it was thought of as research tools, and the impact these have had in a variety of care settings has really been a delight to those of us who were there at the beginning of this effort. And I think this is an area which will strengthen capture of important variables for emulation or trial mimicking strategies. But right now, this is used generally in very local environments, in a pain clinic or in other setting or in cancer, but isn't captured generally into larger repositories of data. And certainly, PCORI has recognized this as a real strategic imperative going forward. Our name is patient-centered, of course, and we really want to see this aspect of electronic health record keeping get stronger. Maybe that's a start on the topic.
No, I think it is a start, and I don't know if any of the other panelists want to comment on this. You're right that these measures have mainly been used in particular settings, including in clinical research or effectiveness research. I do hope, and I think that's changing. For many of the conditions that we care about in healthcare, these functional measures, which are really patient-reported at the core, are the most important thing. Take degenerative joint disease and treatments that include joint replacement and other alternatives. People are really focused on what is the impact on my function, my ability to do what matters to me. And we have started seeing in some of the alternative payment models a focus on finding the best way to improve functional status. Not necessarily just paying for the joint replacement procedures. So hopefully that will get more systematic. But I think the more that we can reinforce the validity of these measures through other systems, payment and the like, would help. Any other comments before we leave the topic of data on where we're headed? It does seem like a general trend, like we've seen in Sentinel, towards more inclusion of electronic records, more efforts. While this is hard, while what we've talked about is hard, more confidence that bigger data sets can be brought together if there is all this work on curation or just understanding the data, for lack of a better term. Any other comments on where we're headed, or advice for moving the data validation and efforts to assure that more data are fit for purpose in observational studies, moving those along?
So just one other comment about the patient-reported outcomes. Some of you may be familiar with the My Studies app, which FDA has made available. And that started as actually an app in Sentinel. And its first pilot project was to try to collect data from pregnant women that wasn't available in the claims or the medical record. And so that's another approach. So if the patient-reported outcomes are not incorporated into patient care, but it's felt to be critical, they can be collected. Now, it changes the dynamics of the study somewhat, because you're now interacting with the patients, and it's not just retrospectively looking at the data. But it is an alternative approach that's probably worth keeping in mind.
Michelle, were you going to say something? It looks like
I just have two quick thoughts. One is that the capacity to access and analyze vast stores of data has rapidly increased. But at its core, if we don't understand how those data came into being, we're at great risk of making inference about the results that really is not supported. And so I look at the N3C data, for instance, which I think is a really exciting opportunity to try to understand COVID. But the data generation process that put those data together is somewhat different by each site. And if you haven't looked carefully to see, oh, it turns out that site X isn't reporting any mortality data, and just sort of forge ahead, you can really miss key pieces of information. And the other side of that is we're also creating new data silos around COVID that really concern me. So the vaccination process has led to different streams of data to collect information on who has had vaccine, which vaccine, when they got the doses. And as best as I can tell, these data are not currently linked with claims or EMR data, which are really our best shot at trying to study safety. So I think we'll get there in the long term, but in the short term, we have some new sort of data silo problems that I think we need to think about addressing.
Thank you all. And just one more before we leave data. A couple of questions related to the same issue of can we be doing more to validate better endpoint data. And Bob, this is directed to you, but may not, but I think if others know about it, there's a new CEDAR initiative, iSTAND, related to validating real world endpoints. Does anyone want to comment on that, or any other FDA initiatives on real world endpoint validation? Okay, well, so suffice to say that there is more work going on in those areas. Let me shift gears to talk about methods. So that next key piece of the FDA framework on real world evidence, and something that many of you talked at length about, having well developed and well understood methods for population comparators. For Josie, I think you highlighted the value of looking at trial analytics and applying some of the principles here, and Michelle, others highlighted that, well, you need to do that and more because of the additional issues that come into play with observational studies, like determining time zero and things like that. Are we there yet in terms of, a lot of the questions in the chat seem to be looking for, well, is there a tool book or guidebook or best practices I can use for a particular kind of application? Anything that you all want to say about this? And maybe Lucinda, could I start with you? As I know ISPOR spent a lot of time on trying to identify best practices in these areas of methods. And as we heard in your initial comments, it's not just about residual bias or confounding bias, but addressing a lot of these other issues well. Which my takeaway from the talks yesterday was that while important to think about, these are issues that can be addressed maybe a lot more effectively than they generally are.
Yeah, I mean, I think it depends on the underlying data and what the question is that you're trying to answer, and not trying to run to your favorite Cox proportional hazard model because that's what your favorite methods, but really trying to think about what you're trying to do with the data. And I don't think ISPOR or ISPE, for that matter, because we collaborated on some of these, has been that prescriptive about exactly how to do the analysis, but to think carefully about the question and then apply the best method to try to answer it. That doesn't lead to any sort of checklist or toolbox where you can pull out. And I think actually moving away from some of those, just thinking about my favorite hammer or wrench to answer the question, but think really critically about it, and maybe even collaboratively. So if you're not sure, thinking. Again, maybe going back to a clinical trial, the biostatisticians, epidemiologists, there's different ways to answer these questions. And I think, thinking flexibly about it is probably the best answer.
Okay.
Yes, I would add there that I'm not a trialist, but I think there are some really well understood standards that can be put in place to ensure that a randomized trial is conducted in a way in which we minimize the types of biases that we would worry about. And observational data is, there isn't one way, and it depends on the question, and it depends on what comparators you have to work with. And, this is why people do an entire PhD level set of training. So I think it is much more about bringing in that expertise as part of the discussion around how to get there, and to think about what are some design options that you really need to think about, and what are the potential biases that you might be introducing if you don't do it this way. And some of the concerns that drive trial design, as I was saying before, about well, we need the sample size, we can't throw away those people, are actually secondary in this setting, because if we get a highly precise biased answer, that's actually worse than getting no answer.
Yeah, Michelle, and just to follow up on, you mentioned this earlier in your comments about the need for, so not only some expertise on the design of the and application of the methods, but also different expert perspectives. You mentioned statistician perspective, pharmacoepi perspective. Can you say a little bit about how to get those to work together well in a good observational analysis?
So I think bringing people together early, and as Miguel highlighted for us, getting real clarity on what is the question we're trying to answer early on. So I'm sure the statistician would tell you, like, we really would rather that you didn't call us at the point when you're trying to do the analysis. We'd really, really like to be involved a bit earlier. And I think because so many of the decisions around study design are critical, you really do need this team in place early in the process. And you can't get away from the fact that this is a team science type activity. You have to have people who understand the clinical context, people who are understand the data generation, people who can think about the study design, given both of those. potentially statistical expertise to think about how are we going to analyze this, especially if it's a time varying or a chronic treatment. The methods that we use for point exposure type treatments just aren't going to cut it, to be honest. So it is a team effort, and it needs to start at the beginning.
All right. Other panelists want to comment on this, Josie or Bob?
Go ahead, Josie.
Couple thoughts. I think immortal time bias was something I was personally educated on in the last few months, and it is an important source of potential error. Once you get that, that's something that can be addressed pretty straightforwardly. I do think that there are other risks that don't go away quite as easily. And propensity score matching has also become pretty much the norm. What I learned yesterday really was the value of having a good comparator as a way to identify a reasonably comparable matched group. And I think those three things together are my sort of short list in thinking about whether to look seriously at an observational treatment comparison. Have they dealt with immortal time bias? Have they done good propensity matching? And have they got access to enough variables to do that well? And then is there a reasonable comparator? The remdesivir example, which I cited, and we're still struggling with a couple of papers on this, there hasn't been a good comparator. So there hasn't been a good way. First of all, the risk of the outcome is very common, but nobody's come to us with a study in which they have a good comparator approach that convinces me that the bias in remdesivir being given to the same people who are at risk for acute kidney injury, is reduced.
I like that.
Non-statistical approach to what we were learning.
Yeah, I like that list of three key areas. Bob, anything to add to this discussion around finding the right methods?
Right. I wanted to pick up on a point that Michelle made, I think, in her slides, and that is the use of sensitivity analysis or quantitative bias assessment. And I think it's very important. Typically now it's done after the fact, but I think it can be also be incorporated into the design phase. Once you've done some of these fundamental issues, got the fundamental issues around design straight, if there's still questions of, is there an unmeasured confounder that I'm still worried about? Well, you can do a quantitative assessment of that potential impact relative to the effect size you're trying to study. So incorporating these ideas, I think it will be very helpful.
Yeah, and just to pick up on that, there was a question in the chat about recognizing the importance of sensitivity analysis. But if you basically the question was the effect of, well, if you're concerned about unmeasured bias and you don't know how big it is, well, what kind of sensitivity do you use? Any comments on good practices there, Michelle or others?
Sure, I think I tried to answer that in the chat, just to say that one of the nice things about a quantitative bias analysis is it really can be applied anywhere from a setting where you only have a sort of a nested validation study where you have some additional information on a smaller subset of the study population that you're looking at, all the way to using the investigator's sort of best estimate of plausible associations. And you can explore a whole range of values that you think could potentially be in play. And then you can look at your treatment effect estimate accounting for those potential differences. And you can even get a confidence interval that's adjusted for that accounts for your uncertainty about those parameters. I will say at the end of the day, all of that can be a little bit daunting, not because it's hard to do, but because the precision of your estimate is going to be affected by that uncertainty. But if you don't do it, you're essentially assuming that you have measured everything perfectly, that there is no unmeasured confounding, and that you have no additional variance that needs to be accounted for in the estimate. So I think there's a lot of value in it. And I guess another call probably to the journal editors among the audience is this is eminently doable. If we don't ask for it in the peer review process, it won't be done. So we're going to have to demand it or it won't show up.
Yeah, and maybe this is a related question before we leave these issues of methods and residual bias and go to, okay, so what do we do from here to encourage more of these best practices, like the advice for the journal editors? I'm paraphrasing and simplifying a little bit, but what I took from some of yesterday's discussion and today's, and I'm not sure Bob Temple would agree with this, but if you take care of the factors like Josie was just mentioning, you know, making sure that you have a good comparator and that you've matched the population well, and taking care of immortal time bias and these other steps like we've been talking about, maybe unmeasured confounding is not likely to be a big problem. But how do we really know? Is the sensitivity analysis, is that the best way to use as a check? Because again, we're kind of dealing with things that are, this is the residual unmeasured.
So, I mean, this is sort of the central question of the DETECT project that I gave you some highlights from the first phase of that work. And we are explicitly trying to compare the different tools that are out there to say, in a controlled setting where we know what the truth is, and we induce some unmeasured confounding, which of these is going to get us the best handle on the risk from that? Again, I don't think the problem is that we don't have tools. The problem is that we're not using them. And we can, you know, again, I hope that this work is going to give us sort of an evidence base from which to say which of these seems like it's the most useful, or which among them are all useful. But I think the threats that we talked about earlier in terms of finding not just a comparator, but an appropriate comparator. You know, if you're comparing a first-line drug to a second-line drug or a third-line drug, you may have a comparator, but it may not be an appropriate comparator. And the really critical factor that comparator does is it identifies in the best case scenario a group of patients who are all at a similar treatment decision point, and where information has been collected and hopefully recorded in the electronic health record or the claims data about where they are at in that disease process. And then we can compare the outcomes, you know, over some follow-up time for those two groups. But the comparator is not just about sort of having a placebo, but it's about anchoring people in time at a similar point in the disease. And whether that is initiating first-line therapy, switching to a second-line treatment, augmenting their regimen, all of those nodes are treatment decisions. And in the case of the remdesivir example, the problem is that we need to find people who look sick enough that they could be getting remdesivir, some of whom are and some of whom aren't. And figuring out what those markers are is actually harder than finding, if there were another treatment, people who initiate some other treatment for those who are really sick with COVID. So the ability to implicitly control or balance those groups using the active comparator is where the real power is.
Great. I see some head nodding. Any other comments before we move on?
Yeah, just one comment. You know, Miguel Hernan yesterday in his example of how the paper was rejected because there was unmeasured confounding, unspecified. I think the conversation would move forward a lot if that was not an acceptable answer, in the sense that if the last thing left is, I think there might be unmeasured confounding, I think there should be some requirement to say, okay, well, where do you think that unmeasured confounding might lie? So you've got this propensity score model with all these variables in it. You think there's still something left. that would be of sufficient magnitude that isn't correlated with any of those variables that could result in this, bias, could you please at least tell me what you think that might be? Because I find that often in these discussions, it's just a knee-jerk, there's unmeasured confounding, so I can't accept this. That would be a huge step forward.
That's a great point. Actually brings us to what I'd like to talk about next, which is how do we solidify more of these emerging promising practices. And several of you brought up the topic of, well, what should, including you just now, Bob, what should journal editors be asking for, and what should we expect of the journal editors themselves? And during the course of this session in particular, and what we've heard earlier, several themes emerge. One of those is transparency about methods, transparency as much as possible about data. I'd like to get back, there were a couple of questions related, well, how much data transparency is feasible and should be expected. Josie, you had a couple of suggestions for the journal editors as well. I guess another theme that came up was pre-specification. So can these approaches be written down ahead of time? And I know, Lucinda, you've done some thinking about how best to handle that. So maybe we could talk a little bit about what's at the top of your wish list for getting the publications, the research done right to support these regulatory applications and hopefully making for a clear path forward. I'd like to hear from each of you.
Well, I guess I'm to go first here, but I do find that some of the standards that have evolved for clinical trials have been very helpful to journal editor decisions. We look at clinicaltrials.gov right away. We say, are they really sticking with their pre-specified analytic plans? And there also are growing standards that the data sets that are used to develop the analytic data sets, that there should be appropriate provision that qualified people can look at those. And we set those data sharing expectations for all the work we accept. I was smiling as Bob was talking, because I realized we make decisions, unlike the FDA, who usually has to tell people something about why they decided. Journal editors don't actually select. We turn away 9 out of 10 papers and don't always exactly explain why, which is, I'm thankful that we're not, that the expectations aren't that we should always do that. And certainly, it isn't uncommon to say we're very interested in this question, but we would rather see published rigorous randomized controlled trials on this question. And that's, something I don't think is going to change immediately. I will add that guideline writers have similar standards. And so the people who are doing this work are going to have to develop and articulate the kind of standards that bring this level of evidence up to what we've all learned to view as our gold standards. I don't really know what to say to an observational researcher about what data set should you be prepared to share. And I'd be interested in some approach to this that would help make this practical. Perhaps Yale repositories, I think, represented in some of this. Perhaps they have ideas on how this can be done. But I think this, and then the second is, should we be requesting pre-specified pre-registration of analytic plans? When I first took over editing this journal, I proposed doing that. And I will tell you, my observational researchers that mostly submit to us were up in arms, and they told us that it's different than clinical trials. We can't necessarily fully pre-specify what we're doing. And Miguel yesterday noted that it's not like the data isn't already there. So people can look and kind of have some sense of where the promise is. So I don't have an answer about pre-registration. It does make the editing peer review process much easier.
Great. Thanks, Josie, for that very thorough answer. So I do want to hear from the other panelists, and we are running a little bit short on time. So Lucinda?
Yeah, so in terms of pre-registration, that's something we are trying to advocate, especially for these types of comparative studies, knowing that there are issues with this, but there's no reason why you can't think about what your hypothesis is, what your analysis plan might be. It could morph or change over time, but there's certainly ways to track the versioning and reasons for any change or deviation. And we've talked to our friends at clinicaltrials.gov, and they said, they also had trouble having uptake of people registering clinical trials until, and I'm going to murder the acronym, the ICJME. So until the medical editors really incentivized it by requiring it, it didn't really happen. Now, I know, again, we can't incentivize something that we need to sort of walk people up to the edge a little bit to get them used to doing, but I think trying to get people to think about their study ahead of time. What is the study they're going to do? What's the analysis they plan to do? And then even have a place where they can put the results at the end, because not everything gets published. And it is a place for people to actually put the results in a way to increase the totality of evidence. So we are advocating for that, knowing that there are issues around some of this, but I think there's ways that we can still think about pre-registration.
Great, thank you. Michelle, Bob, any final thoughts on this topic?
I guess I think we've covered sort of the pre-registration transparency, which are both really important issues. And I hear the call for some guidelines or a checklist or something that's going to let us sort of go through a set of checkboxes and say, yeah, this looks like a good example of RWE, a non-interventional study. I wish I had one to offer you. I think we can move in that direction, but I don't think it's ever going to be as simple as a checklist. It's probably going to be a set of considerations to think about in the context of this specific question. And I think that could be incredibly valuable, even though it's not going to be quite as clean as saying, oh, we're trial participants blinded to their exposure, we're the people assessing outcomes blinded to the exposure. But I think we can move in a direction to provide more helpful guidance to reviewers and editors than what we've done yet.
Thanks. Bob, any final thoughts?
Yeah, just I think picking up on what Miguel Hernan said again yesterday, the idea of for the set of observational studies that are focused on comparison for either safety or efficacy, the idea of pre-specifying your target trial, I think that's a really fantastic place to start. And I don't think it's very commonly done right now. And we go a long way to addressing some of these issues. There's still a whole set of questions once the trial is pre-specified that you're targeting. There still needs to be data exploration to understand how, in fact, it maps to the real world. And there will be some challenges there in the transparency around that. But I think that the trial pre-specification, target trial pre-specification would be another big step forward.
A nice practical, sounds pretty feasible step. A nice one to end on. I want to thank you all for a great and wide-ranging discussion. We got through a lot of the questions. I know we didn't get to a lot of them either. Have another chance with our next distinguished panel. Right now, after thanking our current panelists, I'd like to thank our audience. We ran a little bit over here, so we're going to start the next panel at 2:45 rather than 2:40 Eastern time. So 5 minutes later than on your agenda. So we'll be reconvening in about 10 minutes from now. Thank you all very much. All right, I'd like to welcome everyone back for our final session of this workshop. And also thanks to those of you who are joining us today. We've got a lot of not just questions, but also comments and a bit of dialogue going on in the Q&A. So I appreciate that. It's a great way to augment what we're able to cover in our limited time together. So please keep that coming. Right here, though, we have one more set of expert panelists to synthesize some of the issues related to observational studies to inform regulatory decision making, and also next steps for filling in some of the gaps in the knowledge and the use of real world data and real world evidence that we've identified so far. So we're going to focus on some of the next steps for the role of observational studies in contributing to evidence on drug product safety and effectiveness. Touches on a lot of the topics that have come up in the Q&A as well. And the panelists are going to help us identify some of the gaps and also hopefully help us identify some of the strategies for filling them. We've got a great set of speakers to do that. First, Nancy Dreyer is Senior Vice President and Chief Scientific Officer for Real World Solutions at IQVIA. She's also Adjunct Professor of Epidemiology at the University of North Carolina at Chapel Hill. Frank Harrell is as a Professor of Biostatistics at Vanderbilt University. Rob Reynolds is Vice President of Epidemiology and Patient-Centered Outcomes at GlaxoSmithKline. And John Concato serves as the Associate Director for Real World Evidence in the Office of Medical Policy in the Center for Drug Evaluation and Research at FDA. So Nancy, let's start with you.
Thank you very much. Hope my sound is coming through, guys.
Just fine.
Thanks so much for having me. It's been a really interesting discussion and given me lots of food for thought. But, you know, I started thinking back about Seb's comments, right? So the first day he posed yesterday, he asked two questions to help us sharpen our focus. Can real world evidence estimate causal treatments in a given study? And how could we calibrate the real world evidence against a true gold standard? Now, for this work, the randomized control trial was assumed to be the gold standard. And they selected trials that they thought had a good chance of being replicated. Now, holding that strategy aside, some were replicated and some weren't. So what have we learned from this? If you permit me, I like to play the devil's advocate for a moment. We know some of the problems are the outcomes. Well, I think we knew a lot about that already. We know that the typical practice of medical care is different from idealized trial settings, and that patients don't, for example, they don't always get their glucose monitored with the regularity that you would expect. We also know that unlike in clinical trials, in real world settings, biopsies are ordered when cancer progression is suspected, not as a tool for monitoring progression. So the data is different. We already know that at the outside. We already knew that many of the trial inclusion and exclusion criteria aren't available in real world data because they're not things that are typically measured. And we knew that exposures of interest aren't always recorded or accessible in real world data. Well, we know that what we saw here was studies that were drawn from existing data, not study-specific case report forms. And we know that you could learn more through record linkage or through supplementation with direct data collection or with the use of connected devices. We also heard about the lack of follow-up because people change health plans. Well, we know that. I don't think we talked specifically about the fact that some of the outcomes of interest are not necessarily captured in those health plans. I mean, for anyone who's done mental health research using health insurance claims, you know that the mental health claims can often be an insurance carve-out. So if something happened, it wouldn't be in the database because they don't pay for that, someone else does. That brings the important topic of provenance, Mark, and I think we need to talk more about data sharing. But just to finish up my comments, we also saw different patterns of use of treatments in real world evidence. For example, different age distributions, no matter how hard we tried to match the RCT. This is not uncommon, and it's got to serve as a reminder for us that treatments after approval are used in different populations. So when all is said and done, when we replicate a trial, we congratulate ourselves. When we can't, we look for explanations. But the question is, what enduring tools have or lessons has this given us? You know, we've been working to make the real world evidence match the trials, but it's important to remember that target trials, even elegant ones, often can't be conducted and are never conducted because they're too complex or the subjects are too rare or they just don't exist. I mean, we are often asked to test trial protocols using real world evidence to find out the impact of each inclusion and exclusion criteria so we can understand how much each one reduces the eligible target population, which helps differentiate between a trial that can recruit and can finish and one that never does. So I think the basics here are that a study design doesn't guarantee truth. If randomized controlled trials were simply the ultimate gold standard, we'd never have trials that disagreed with each other. So it seems like we ought to be spending our time focusing on the complementary value of the real world evidence and the RCTs, rather than duplication, replication, or emulation. I mean, we could go back and ask RCTs to be designed to give more real world outcomes, and we would be able to do this evaluation better if that were true. But we know that's an unpopular option because the RCTs often measure intermediate or surrogate outcomes rather than real world events like mortality for good reason. It takes too long to find out the mortality outcome and to see if a product is beneficial. So we can look for real world populations that have the requisite data, but we're learning how unusual that is and how limited and distorted those populations be. And we can add our elegant analytic approaches, but they often have a bit of a black box element. And we've heard today from journal editors saying, how can I evaluate that? And we heard about the Surgisphere, which looked like they did everything you could ever want somebody to do to have high quality evidence, till you started looking how closely at the words they said and wondering, you know, how can they go from last patient in to publication with extraordinary quality control all in 6 weeks? I mean, things like that make you wonder, but only when you're familiar with how these evidence are assembled. So I'd like to get back to basics and remember why we're interested in real world evidence. It adds the generalizability we don't see in the trials. Real world evidence can provide context for single arm trials. We know we can only study marketed products and treatment effects in those settings, but we can certainly use real world evidence for label expansions just because we see how differently products are used in the real world. And we can confirm or rule out relatively large effects for safety or for comparative effectiveness. So it should serve as a reminder that we benefit from looking at the totality of evidence. And we need to look for, as always, we need to look for consistency of findings across data sets and across analytic approaches. That's how science advances, conjecture and refutation, just as Karl Popper postulated in 1963. Seems like we all are starting to agree that real world evidence has value. And when you hear enthusiasm from Bob Temple, you know this is a new era.
Thanks, Nancy. Great point to end on. And next up is Frank Harrell.
And if you want to start my video camera, maybe. Okay, here it goes. Well, I want to continue along with Nancy and being a bit of a devil's advocate. And I'm going to turn mainly to methodologic considerations. But before that, I just want to address a couple of the big picture items as she mentioned. I think I would challenge the group to really start the discussion with a burning question. I don't think we really heard any burning questions in this really excellent conference. What really needs to be answered? What sort of questions are we having trouble answering, especially having trouble launching clinical trials about? How long does it take to do a good observational study? Does it sometimes delay doing a randomized trial so that you can do an observational study? I think we have a pretty classic example with the mess surrounding convalescent plasma observational research. I know FDA was one of the sponsors of the large project that was run out of Mayo Clinic. And not only do we have an uninterpretable result about the efficacy of convalescent plasma, where we actually saw a lot of the nation's convalescent plasma supply used up to do the observational study, and that really hindered randomized trials. That's a lose-lose situation. I think we haven't talked enough about sequencing of research programs. When do you launch an observational study? When do you launch a randomized trial? There needs to be a little bit more strategic planning. I think that randomized trials are much harder to mess up from an analytic standpoint, except for missing data. That's one place where you can mess up. It's so much easier to mess up an observational study because the analysis isn't unique. The time zero maybe isn't unique. And so the training of researchers that are involved in observational research, I don't know where that really stands right now, but I see opportunities for improving methodology with some maybe more systematic training or a little more uniform training. I think the metrics for judging replication are not there yet. And I was concerned with what Sebastian Schweiss presented yesterday, looking at confidence intervals side by side. We know from statistical theory, that's exactly how you don't do it. But you need something like a confidence interval for the amount of disagreement between the two sources, or what a Bayesian would say is we need a posterior probability that the real world evidence disagrees more than 10% with the efficacy estimated in a randomized trial. So I think there's still opportunities to do better quantification of the concordance or agreement between the two types of studies. There are major disadvantages in big data. This isn't talked about enough, but big data, being able to curate the data and to have satisfactory quality control. So I see some occasions where having larger and larger data is actually a disadvantage. I am very concerned about the overuse of propensity scores throughout this field. I really don't understand it. I think it's very problematic. And the most problematic aspect of that is using matching techniques. So I've seen many high-profile observational treatment comparisons where more than 2/3 of the patients in the study have been removed because they weren't used in the matching algorithm. And they weren't removed because they weren't good matches. They were removed because they were good matches that didn't happen to be used by the matching algorithm and the sequence of observations and how it processed the data. There's also a big misunderstanding about how do you look at balancing after doing propensity score matching. This is really a superfluous analysis, because if you fit the propensity model correctly, it has to be balanced. So the fact that we're seeing some examples of some covariates not being balanced, that's just a reflection of mainly assuming a linear relationship inside the propensity score that should not have been assumed. So I see too many investigators spend a lot of time on this, and just in general, propensity scores are being used when you don't need them because the dimensionality of the problem is easily handled with regular covariate adjustment and other standard modeling techniques. I think there's a lot of other methodologic issues should be covered, but I think we need to think more in the way that Nancy was talking. How how do you complement each other? Instead of looking at duplication, can we learn more about treatment interactions from observational data? Can we learn more about the effect of it at varying adherence? And I think that the idea of when to worry about unmeasured confounding is still pretty much of an open question, even though Miguel gave us some compelling examples where that's not the driving issue. In a particular medical question, you really don't know if it's the issue or not. So I hope that irritates just the right number of people.
Thanks, Frank. Rob, you're up next.
Okay, thanks, Adam. So thanks for the invite for the to be a participant in this panel. I wanted to start with a recap of what I thought was our starting position yesterday. What I heard from Jacqueline was that most stakeholders feel comfortable with using real-world data to generate safety RWE, but when it comes to claims about efficacy or effectiveness, we have really a trust and credibility problem that is persisting. That RWE is often viewed as a black box and real-world data as easy to manipulate or game our answers. I think I also heard that we need greater certainty if RWE is to be part of the substantial evidence standard for regulatory decisions. And I, like others before me, was really happy with the progress that we made in working through some of those issues during the session yesterday and today. And being the first being that the target trial framework is clearly a pragmatic approach that one can use in many different settings. It structures our conversation so that we can have a conversation among people who are proponents of RWE and trialists, whether or not they're skeptical of RWE, but it really focuses us. And my own experience with an industry, I've found it to be quite valuable to start there. Along with design issues, which are obviously key, I think we've been hearing through the panels that sensitivity analyses and quantifying potential bias are really important tools to characterize uncertainty. So they increase confidence by decision makers because they can understand the impact of different design specifications, different endpoint definitions. And to me, those are often quite reasonable, some of those different definitions and specifications, and they can really address the question of gaming if we pre-specify transparently the primary and sensitivity analyses that we want to do. And just to pick up on the point from the panel before, I think that often when we're planning for a study of a drug's effectiveness or safety, we're doing so prior to the data having actually been in the database. So it is possible that these things can be pre-specified prospectively without the possibility of having looked in the data beforehand. And then finally, for me yesterday, what I heard from the projects is that, that in general, if we have the right data and we use a proper study design, that we can come to the same conclusions as RCTs, and therefore observational studies can be credible for causal assessments. So like Nancy, I think what I heard from her and what I experienced is that the credibility is improving, that people's view on the credibility is improving. And this really raises for me two questions. The first is, how many replication efforts are enough? Nancy and Michelle have brought up some of the sort of limitations of this approach, and I think we have to ask ourselves, once these projects complete, is that sufficient? The second question for me is, how much credibility do we need? Do we have enough credibility? And I think we do at this point. And so the question becomes, where are the areas where we're ready to trust the credibility of observational studies? And I think there's at least two areas. The first is in the use of external comparator arms or external controls. We heard from Bob Temple yesterday that control arms outside of a clinical trial can be credible, especially when effect sizes are large. And sponsors are interested in this question because, of course, there are many programs where we're developing around indications in oncology or rare disease. And in rare disease, historical controls, of course, have been used for many years, but in places like oncology, where the treatment paradigm is changing rapidly, we often need concurrent or contemporaneous controls. And that's a place where we need to look at RWD or existing clinical trial data and use the target trial framework as a way to decide which is the appropriate external comparison data to use in these situations. And we also need to do that when we're designing the single arm trial. So that means this needs to occur very early in the design phases. We can also think about using real world evidence to augment trials that are randomized and have controls. So we often have controls where we don't have very many of them, or we have very short follow-up. And in those instances, I think we're ready to really explore the use of real-world data, either through secondary databases or through EMRs, registries, et cetera. There's really a lot of options that we can take advantage of today. I think the second area that's really important is in supplemental indications and labeling extensions, and that includes pediatric populations. This is an area of focus for 21st Century Cures and PDUFA 4, and to me it seems like a sweet spot. And the reason for that is because a lot of times we cannot do trials in these areas. They're either difficult to enroll or there's no equipoise, and so we're really facing an option where we can either not do a trial, or we can design a proper real-world evidence study that emulates a trial and have evidence to drive decision making. And then finally, I want to just make the point that sort of in order to really advance the use of these approaches within drug development and as part of regulatory decisions, that sponsors do need clarity on some of the expectations and guidance. And so conferences like this, of course, help that. But we do need to be able to advance use cases, specific use cases that will help us develop clear principles over time. And that is going to require early engagement for sponsors with regulatory agencies, and I think increasingly payers as well, because payers are grappling with many of the same issues that regulators are in terms of thinking how to use real-world evidence for reimbursement. So those are my comments. Thank you.
Great. Thanks very much, Rob. And next we're going to go to John Concato.
Thanks, Mark, and thanks to my co-panelists. I'll share my perspectives in categories of what we knew already before this meeting, what we've learned from the replication analyses, and where we go from here. If we focus on methodology, and as you've heard, we've known for more than 20 years that despite all the strengths of randomized trials, results of trials conducted on the same clinical topic themselves don't always agree. We've also known that the results of observational studies are sometimes wrong and sometimes right. And as Frank just mentioned, there are various ways for an observational study to get the wrong answer. Yet, when considering this issue, we sometimes oversimplify the situation by invoking a hierarchy of study designs before objectively evaluating the strengths and limitations of a particular study. The complexity of evaluating evidence is highlighted by the example of hormone replacement therapy and cardiovascular outcomes that Miguel Hernan mentioned yesterday. Rather than RCTs correcting flawed observational analysis, which is still a misperception out there, the RCTs and observational studies were actually answering different research questions, and the process involved also confirmed that new user designs are important in observational research. So the bar was raised, or the floor, I should say, was raised in terms of conducting good observational research. This example and others highlights how RCTs and observational evidence can combine together to enhance our understanding of a drug's benefit and risks. Now, from a regulatory perspective, we have decades of experience at FDA evaluating evidence from randomized trials, whereas evaluating real-world evidence for effectiveness, at least, is a relatively new challenge. I say relatively new because epidemiologic terms for various study designs were in common usage before the terms real-world data and real-world evidence were popularized. In fact, FDA was approving what we now call real-world evidence prior to the passage of the 21st Century Cures Act, as with externally controlled trials called historical controlled trials in our regs from the 1970s, as Bob Temple mentioned yesterday. So I would mention it could be said that in addition to innovation and new ideas, even terminology has not yet been socialized in the era of big data. Regarding what this meeting added, and a lot has been said, I agree with virtually all of it. Quite a lot has been accomplished, as we've heard from prior speakers. Within FDA, I'll mention that CDER and CBER are supporting the evaluation of real-world evidence with a four-prong approach: internal agency processes, external stakeholder engagement, demonstration projects, and guidance development. RCT duplicate is one of our more prominent demonstration projects, and FDA thanks the team based at Brigham and Women's Hospital for their meticulous approach to this work. We also thank the OBrien team, the El Mayo CERCI, Duke-Margolis, the meeting's planning committee, and all presenters and panelists. So the replication results overall that we heard about yesterday and we've talked about today can be characterized as adding to a body of work that advances our understanding of how to conduct rigorous observational analyses, in this case using claims data. For example, we're reminded that a good number of design factors can influence the result of trial emulations, and just thinking about the permutation of those factors adds up quite quickly. But in general, with more experience, we can learn what works and what doesn't work in terms of generating valid results from observational studies. But even if we agree on best practices for observational research from a methodologic perspective, the regulatory requirement for adequate and well-controlled studies to approve a drug remains the threshold to be achieved. Although observational studies have upside potential, the replication results show that we need to learn more about RWD and RWE going forward. There's certainly no easy answer in terms of a checklist for fit-for-use data, an off-the-shelf study design, nor an ideal analytic method. So what's left to discuss is where do we go from here? And I'll start by emphasizing that FDA is embracing our obligations under the 21st Century Cures Act to evaluate the use of real-world data and real-world evidence. As a simple action item, I would say we could all be a bit more disciplined when using terminology. For example, the term observational study suggests a cohort or other design architecture to evaluate a drug outcome association. But observational study is also sometimes applied, it seems, to any descriptive analysis that involves real-world data. So when we have the right vocabulary, I would advocate we shy away from the false dichotomy of randomized trials or observational studies, and instead allow ourselves to think about when and how randomized trials and observational studies can be designed and conducted to yield trustworthy results. Taking it a step further, and although we don't have time to go into details today, interventional and non-interventional studies is arguably a better taxonomy for study design than RCT versus observational study. We should also recognize and adhere to principles of rigorous science, as we've heard today, when planning and conducting observational, I should say non-interventional studies, with the science involved being clinical epidemiology. As mentioned in FDA's real-world evidence framework, fit-for-use data, adequate study design, and appropriate study conduct from a regulatory perspective are the basis for the agency evaluating real-world evidence. In turn, sponsors should maintain high standards when evaluating these criteria and not try to justify, for example, a real-world data source simply because it's available or a list of covariates because they happen to be in a data set. As we've also heard today, sponsors have the responsibility to plan and conduct non-interventional studies with transparency and with integrity, including submitting a protocol before conducting any hypothesis testing analyses. We have more work to do in this space, but we're off to a good start. So in closing, we heard yesterday that the target trial approach and related ideas actually have been available for decades and yet have been underused. So perhaps we're approaching an inflection point regarding how observational studies are generated and how they are viewed. We'll only know whether that's true or not in hindsight, but FDA is committed to help improving the quality of real-world data and real-world evidence. Thanks, and back to you, Mark.
Great. Thanks very much, John. Great comments from all of you. And I'd particularly like in our last few minutes to pick up on John's looking forward from here. Potentially progress would have been possible decades ago. It's nice to be seeing so much progress happening now, building off the target trial model and the work, the demonstration work that FDA has supported and that so many of you are engaged in. It does seem like we're headed towards, in the keeping with the FDA's spirit of totality of evidence approaches to analysis, more integration. So Nancy mentioned this in her comments that replication is useful, but that's not where we should stop. There was some discussion yesterday about replication potentially being a foundation for addressing longer term outcomes, other populations, things that can't be handled easily in a feasible clinical trial framework. I heard from some of the comments today from Rob and others about ways to combine clinical trials, and John, I appreciate your use of the terminology, non-randomized, randomized studies, and really more comprehensive approaches to analysis. John, maybe we could start with one quick question with you, and to give the rest of the panel a chance to think about where we go from here to build out this more integrated approach. One of the things that Rob mentioned was, if there are these opportunities around label extensions in rare diseases where you're building on effect sizes or examples and past practices at FDA for using non-randomized studies or external control arms. Are there more steps you see coming as the FDA works on its obligations, and they're doing a lot under 21st Century Cures to support those use cases? I assume the guidance has come to you early, and think about how to fit this into an overall development plan, and maybe engage some of the payers and others who care about other post-market questions as well.
Thanks, Mark. Well, as I mentioned, we have a four-pronged approach, and they all come together. We've been focusing on demonstration projects and one prominent demonstration project, but we have a portfolio. Michelle mentioned another, looking at assessment of confounding. If you put that together with our guidance development, which, they're forthcoming, we published our guidance agenda not that long ago, our external engagement as well as our internal agency processes, all of this is moving together in an integrated fashion to work together to improve the quality and to improve awareness and to come up with more concrete recommendations, recognizing that at the end of the day, two things are true. One is that our evidentiary standard will not be lowered. And two is that it's a case-by-case decision, both on the sponsor's point of view in terms of what they're going to submit and on the agency end in terms of the review process. So that's the tension involved, or the constraints involved. But on the other hand, it doesn't mean that we can't take one step at a time and accomplish the so-called journey of 1,000 miles or whatnot.
Yeah, great. Thank you. Nancy, going to go back to you in terms of where we go from here, especially on integrating the interventional, non-interventional approaches, or this more of a totality of evidence strategy.
Well, I'm going to leave that to the regulators, Mark. I think that we have a good foundation for looking at the totality of evidence. But if I have 30 seconds at the mic, maybe we can talk for a minute about how you evaluate data and data sharing, because I think it's easy to say, let's share the data. And I think we've got our eye on the wrong ball, or we're not really thinking about what we're doing here. First of all, there are all the issues of privacy. When you do a randomized controlled trial, you get informed consent, and you can do a variety of things with the data that you got consented for. When you're using somebody's electronic health record, there are limits to what you can share just because you think it's good scientific practice and it could contribute to science. So it's not necessarily that easy. And I think I just want to emphasize the point that Michelle brought up about provenance, because Mark, I've been arguing with some of your predecessors from the trial world who say that what we need to do is submit to the FDA the real world data that were used to drive an analysis so that they can replicate our analysis and make sure we did it right. But it seems to me you need to start upstream understanding why those data were created, why they came into the data set, and that whole structure of where it came from, why it was recorded, how you transformed it, how you checked it. That's a very big question. You can't submit all the Kaiser Permanente or insert your institution's name here data so you can understand that. So we need to rethink that process.
Great, thank you. And next, maybe if you go to Frank, comments you've got to add. And by the way, Frank, I think your comment about needing more training and support, that's generated some discussion about there are some programs out there that can help, that ISPE, International Society for Pharmacoepidemiology, others support. Your comments?
Yeah, I think the proof of the pudding will be, and I know I would be one who would be critical of anyone's curriculum because I have a specific way of practicing biostatistics, but I think we've learned so much about non-randomized treatment comparisons in the last 5 years that really changes how we do it, and how much of that has crept into the training. I'm not totally convinced we've caught up yet, but I would like to be pleasantly surprised. And just to bring up another angle where I think we need to have maximum complementation of the two approaches, people are doing more difference in difference designs. And I think when you look at double differences, there are opportunities. And in the bigger framework, we think of interactions or double differences. And sometimes we can model interactions when we cannot model main effects. So you might have a confounding by indication, which makes your observational data shifted upward by an amount that you don't know how to correct for. But it may be some relative differences off of that, which deals with differential treatment effect, can still be ascertained and combined with average treatment effect from a randomized trial. So I think we need to seek more ways to complement. And that relates to another way to complement each other, which is observational data, or however you want to call it, non-randomized data, does sometimes have patient spectrum advantages. And where that really comes into play is you can use randomized trials for what they're best, which is estimating relative efficacy. And then you can use observational data to translate relative efficacy into absolute effectiveness. And so it's a wonderful marriage, and we don't see enough of that. And it's one other thing that it gives me an opportunity to say, which is, I think it was probably Michelle Johnson-Funk raised an issue about maybe populations differ between randomized trials and observational data, and that might make us worry about heterogeneity of treatment effect. I don't think that's actually the case. I think that's not the primary reason why you would have heterogeneity of treatment effect. And so we see randomized trials give us efficacy estimates that are far more transportable than people want to admit, but that still doesn't estimate absolute effectiveness in a way you could do by marrying the clinical trial data with observational data.
Great, Frank, thanks very much. Rob, I'll go to you next for reactions to this.
Yeah, sure. Well, so I would again return, I think, to when we're thinking about real-world data, sometimes we get very, I guess, boxed in by starting with the RCTs. So, you know, for example, around endpoints, I think that we often talk about the misalignment with RCTs and RWD as a problem, but, you know, it can be reframed as really an opportunity to better align both RCTs and RWE with endpoints that are more patient-centric and more relevant to clinical practice. So I think that, you know, we kind of need to go both ways when we're thinking about the RCT and the real-world data. And in that sense, I agree with some of Frank's comments about sequencing. And that was in many ways my point about the supplemental indications, is that if you have a pivotal trial upon which you've made an approval, that is a kind of sequencing that you know, might be acceptable, because then you can begin to look at the use of real-world evidence and have some comfort around what you've seen from those pivotal trials. I think the other point I would make about we often seem to talk about primary data collection, and then we want to add in the real world data collection. I think we should start doing it the other way around. There's a lot of rich secondary data that could be supplemented with primary data collection using the new digital tools that we have, and it could be done probably relatively inexpensively, and still collecting information that's valuable to patients and clinicians.
Thank
and
you.
Based on Mark, can I just
John,
John,
please go ahead.
Yeah, nothing I said, Rob, precludes what you were saying, for example, about label expansions and whatnot. And your point about data is very well taken. Other demonstration projects that FDA is supporting, for example, is working towards moving research data in quotes and clinical data closer together, right? A well-known person in this field says collected once, use it multiple times. Yeah, so part of it is we're in 2021 right now with the system we have, but in oncology, for example, they've made great advances in trying to have, as we know, data, say on RECIST criteria, be collected by clinicians so that the quality of the real world data is improved and there's benefits downstream to real world evidence. So part of this is a dynamic process. We're trying to look forward in time, but be reasonable enough not to, and ambitious enough to make progress, but without trying to, hit a home run, so to speak, and swing and miss instead. Thanks, Mark.
I know we have limited time left, but since Nancy pushed on the issue of data sharing, any other comments about supporting exactly what you said, John? Do we need some new policies or new steps related to data sharing?
Yeah, well, the agency is certainly keenly aware this is a complex issue. So besides the guidance development that we're working on, we have point-in-time decisions that we have to make about applications we see now, but we also have to look down the road to say, what do we need to do to enable it? So it's a complicated issue with about 4 minutes left. Even if we had 40 minutes, I don't think we'd be able to, number one, solve it. Number two, I wouldn't be able to say that much, but certainly stakeholders can be or hopefully somewhat reassured that the agency is thinking about it. And even if we weren't thinking about it, we're seeing applications that are forcing us to deal with it. Yeah. So as time goes by, we hopefully can work together to sort things out. But we recognize there are multiple quote unquote tensions involved. And if it was that easy, it would have been fixed already. It would have been addressed already.
Yeah, and we do have only 3 minutes left, but John, I would like to pick up on your other comment about is this potentially an inflection point? Just for a final thought from each of our panelists, what step would you recommend to try to make sure this becomes an inflection point? I do want to mention in the chat, I'm not sure it's available to everyone, some good discussion about the programs that the International Society for Pharmaceutical Engineering has available to support training and best practices in this area, and then some calls for expanding some of these training programs. We've got some good resources in the participants in this event, but other steps. Nancy, maybe we could start with you. And I think you're on mute.
I think I'm off mute now. I'd like to see some guidance on data quality, starting with a reasonable expectation for getting some common descriptions about data provenance and data handling. I think that would be a huge step forward. We heard so much today about having data that was discarded for one reason or another. That's really important. And so I think just starting with the basics about how do you describe your data assets and their provenance and what you did with them, including derived variables.
Great, thank you. And next off to Rob, sorry, Michelle. Michelle pointed out that I did misspeak a little bit. It's the International Society for Pharmacoepidemiology. That's what I said the first time. Sorry, I didn't get it right the second time. International Society for Pharmacoepidemiology's resources. So sorry about that, Frank.
I think one thing I'd like to see is more written about how to use an intentional observational or non-interventional study to lay the groundwork for a better RCT, and how to use an RCT to lay the groundwork for a better non-interventional study to refine what we've learned from the RCT.
Yeah, sounds like a good extension of the target model so far. Rob?
So just say I'm looking forward to the forthcoming guidance from the FDA. I think this is really important. And what I'd like to see is that we're able to have these types of dialogues early in the design of programs, because I think, just from the last 2 days, we've seen how much agreement and possible alignment there may be on some of these issues, and I don't think we would have had that if we hadn't had the opportunity to have this kind of conversation.
Great, thank you. And John, I want to thank you and the other participants in this event from the FDA and all the work that went into doing it. Any final thought from your standpoint?
Looking at the hour, it's already 3:30 Eastern. I'll just say for Rob and others, it'll be a portfolio of guidances, not one Uber guidance. So please have that as part of the expectations involved. Thanks very much, and thanks for inviting.
Okay, great. Thanks all of you on the panel, all of our discussants, and everyone who contributed questions and comments in the Q&A and in the chat. Made for a very lively time together, even though we couldn't be together in person. Looking ahead, as you heard, more coming from the FDA. There also will be some more activities at Duke-Margolis, part of our collaborative agreement and part of our real world evidence collaborative. So stay tuned for to stay connected with us as well. I'd like to thank all of our collaborators at the Food and Drug Administration and the planning group who guided the development of the workshop over the past 2 days, in particular, Jacqueline Corrigan-Correa, John Concato, Diane Perrowan, and the OMP, the whole OMP team for their substantial efforts to shape the discussion today, and the external planning committee as well for their time and expertise. Finally, our team at Duke-Margolis, Adam Kretsch, Joy Eckert, Mara Gill, Trevin Locke, Valerie Parker, Marta Wazinska, Morgan Romine, Luke DeRocher, and Patty Green. It really does take a village to build an effort like this. And finally, thanks to all of you who joined us today. I hope you have a great rest of the afternoon, and hope we are at truly at an inflection point in progress on the development and use of real world evidence. Thank you all very much.